Passive transport means in this transport process, no energy is required, no ATP is hydrolyzed. There are two types of passive transport. One is diffusion, another is osmosis. Diffusion is further subdivided into two types. One is the simple diffusion, another is the facilitated diffusion. So we will start with the first type of passive transport, that is diffusion. What is diffusion? Diffusion means movement of molecules from area of high concentration to area of low concentration. In this picture we can see that at one side there is a fluid with solution with high concentration. So on the other side there is a low concentration solution. So there is a movement of fluid from high concentration to low concentration. So as it is happening along the concentration gradient, so no energy is required for it. Okay, so how long this movement will continue? Until and unless both the side will come to an equilibrium. Okay, so and the time required for the equilibrium by diffusion is proportional to the square of the diffusion distance. It is proportional to the square of the diffusion distance. We know that there are two types of diffusions. One is a simple diffusion, another is the facilitated diffusions. What are the similarities between these two type of diffusion? In both the type of diffusion, it is a passive diffusion, so it does not require any energy. So the movement of fluid will happen from high concentration to low concentration. In both the cases, in case of simple diffusion and in case of facilitated diffusion. So now how actually they differ? In case of simple diffusion, it occurs through the phospholipid bilayer of the cell membrane, and it mainly happens through the non-polar for the small and non-polar particles. This simple diffusion happens. Whereas facilitated diffusion, it requires a carrier protein, a transmembrane carrier protein. This blue thing, this transmembrane carrier protein is required for it. And this type of transport, it is mainly for the large particles or the polar particles. Okay, so the similarity is no energy and both from the along the concentration gradient. But difference is in case of simple diffusion, it is no transport or carrier protein is required, but in case of facilitated diffusion, the carrier protein is required. And in case of facilitated diffusion, the process of diffusion, it is fast. It is faster than the simple diffusion. And also we will see in the next slide, the facilitated diffusion, they follow the saturation kinetics, but in case of simple diffusion, no such saturation happens. We can explain this diffusion, two types of diffusion, simple and facilitated diffusion, by these two simple examples, real life example. Suppose some person are coming down through a slope of a hill. Okay, this on the left hand side, these two person are coming down. On the right hand side, another person is coming down through the slope. What is the difference? They are coming down on the left hand side, they are coming down by walking, whereas on the right hand side, this person is coming down with the help of a skate. With the help by doing skating, the person is coming down. So who will come faster? Obviously the person with the skate will come down faster. So this is the example of facilitated diffusion, and these skates are actually the carrier proteins. They are helping the faster movement. Okay. But what will happen after certain time, when suppose instead of 1% there is 100% and only 10 skates are available. So we will see that although the number of person it is high, so as the available transport proteins or carrier proteins are not adequate, so after certain time there will be a kind of saturation. So in case of the facilitated diffusion, the process of diffusion is faster, but after certain period it will attain a saturation. That is the saturation kinetics. We will see it by the graphical representation in the next slide. Here in this slide, we can say that in this graph, the concentration is plotted along the x-axis, and along the y-axis, the rate of diffusion is plotted. So this green straight line is representing the simple diffusion. We can see as we increase the concentration, there will be simultaneous increase in the rate of diffusion. Okay, but in case of facilitated diffusion, after certain period, we will see that there will be a phase of saturation. When all the carrier proteins or the transported proteins, they are saturated, they are not available. So in that time, at that time, a further increase in concentration on the one side of membrane will not enhance the rate of diffusion. So it will come to a saturated phase. So this saturation kinetics is the specialty of the facilitated diffusion. So what are the similarities between facilitated and simple diffusion? Both are passive diffusion. Along the concentration gradient, no energy is required. In case of facilitated diffusion, we require the carrier protein. Mostly the larger particles and the polar particles, they are transported by facilitated diffusion. It is faster diffusion, but it follows the saturation kinetics. Now the magnitude of the diffusing tendency that can be expressed by this Fick's law of diffusion. The magnitude of diffusing tendency from one region to another separated by a membrane. This membrane may be a cell membrane, it may be a blood glass barrier, is directly proportional to the cross-sectional area across which actually this diffusion will take place. And the concentration gradient, that is the chemical gradient, okay, which is the difference of concentration of the diffusing substance divided by the thickness of the boundary. Now this is the Fick's law of diffusion, where J is equal to minus dA and into the delta C by delta X, where J is the rate of diffusion, D is the diffusion coefficient, A is the area, and delta C by delta X is the concentration gradient. And this D or diffusion coefficient, it will depend upon the particles solubility divided by square root of the molecular weight. Now why the minus sign is given? This minus sign actually indicates the direction of diffusion. When considering movement of molecules from higher to a lower concentration, this delta C by delta X, it will become negative. So multiplying minus dA with this minus delta C by delta X, it will give a positive value. And the permeabilities of the boundaries across which the diffusion occurs, it may vary in different part of the body, but diffusion is still a major force that affects the distribution of water and solutes. These are the references, and thank you.